Hello, everyone. My name is Kristin Schirmer. I work at the Swiss Federal Institute of Aquatic Science and Technology, also known as EAWAG. And here at EAWAG, with my group, we have developed the RTGILL-W1 cell line assay. It is a fish cell line acute toxicity test, which allows you to predict the acute toxicity of chemicals to fish. Now, this test has risen to being an OECD test guideline with the number 249. And I would like to say that I'm really honored and very thankful that I have been given the opportunity to share some of the insights with you that we have gathered along the journey of actually establishing the OECD test guideline 249 and more specifically of its validation. Now, we were asked by how and by whom our test has been validated, and I can proudly say that we organized the validation of our study ourselves. Now, for this, I need to give you a little bit of history because the origin of this assay actually goes back to a dedicated Cefic LRI call, which really asked to develop a cell line alternative to the acute fish toxicity test. And this was always something that I had dreamt of, and I felt this is my call. I wrote a proposal and I won the competition. Then when with my team, we prepared this assay, we developed it step by step, and we ran a proof of concept study, which looked very promising, and from which I was encouraged by the liaison team of the Cefic project, but as well by many colleagues to actually start a validation exercise or an international round-robin test. Now, at the same time, and you see some references here, at the same time, I was also encouraged to submit the assay procedure to ISO. And I would like to say that at that point, we also won an award together with my colleague, Melanie Fisher, whom you see to the second to the right here. And I would really like to emphasize this because without Melanie, if we weren't a team, I wouldn't be standing here in front of you today. And then finally, we submitted also in 2019 the test guideline protocol to the OECD, where it was accepted in 2021. Now, it's quite special because this cell line assay is the first ever cell-based assay test guideline in the environmental risk assessment space. And I would also like to stress that we actually, based on the success of the development of this assay, we started an EAWAG spin-off, Aquatox Solutions, which is now a fully independent company and which offers this test along with other fish cell line assays as a service. But what we are interested in today is this step here, the international round-robin test. So the question is, how was your validation study funded? Were there any sponsors? And here I can say that we were lucky to have some initial funds, namely provided by the Cefic LRI, who also funded the initial proof of concept study, together with the UK NC3Rs. Now, at the time, I thought I have this really luxury budget. I asked for a project manager for 50% for 1 year. And we also asked for support for a chemical analyst who could run our samples at the same place at Utrecht University for 3 months. As well, I had requested some consumables and travel funds. Now, I can tell you that this initial budget was far from being sufficient. So essential funds were actually needed, and I was lucky enough again to gather some funds from EAWAG that allowed the continued support of a project manager, and also the participants of our round-robin study largely financed themselves from internal sources. So how did we design the laboratory management for this test? How many labs were involved? How did we deal with the test chemicals? Now, first of all, we had 6 laboratories from academia and industry, and it was not difficult for us to find these labs. They were very motivated, and so that was a really smooth part of the project, and I'm very thankful for them for their contributions. We had 5 labs from Europe, and one from the US. And this is important because we learned that shipping samples back and forth within Europe is difficult, but going across the ocean to the US is even more complicated. Then we organized the round robin testing in 3 rounds. In the first round, we trained 2 laboratories on site here as the lead lab, which was really beneficial because we were able to further improve the SOP, which was already very good, but like there's always room for improvement. And also importantly, we were actually able to further simplify it. And we also developed a method, a suggestion for proficiency test, where labs can be confident, demonstrate that they actually can run this test sufficiently. Now, once these first 2 labs had established this, we trained the remaining 4 labs in exactly the same way. And then everyone went off and tested the 6 chemicals with some exceptions. Now, what were the exceptions from testing all 6 chemicals that we had selected? One person from one testing lab fell ill and could not be replaced. So this lab then only tested the reference chemical. In another lab, we had difficulties that based on the facility rules, they were not allowed to test one of our 6 chemicals due to its toxicity. So this lab then only tested 5 instead of 6 chemicals. What is also important is that we have a step in this assay which requires you to take samples for chemical analysis. Now, we didn't want to validate the chemical analysis part, so we wanted to go with one single testing lab, which we had in Europe at Utrecht University. But then again, shipping samples across to the US or back was nearly impossible. And so the US partners then offered to do the chemical analysis on site themselves. For the handling of data, we collected here at the lead lab all the raw data. We evaluated the data, including using in-house support for statistical analysis. All right, then there are more questions. How were the chemicals selected and how did you purchase them? So we selected our chemicals based on prior experience, namely based on the long chemical list that we had established for the proof of concept study in the CEFIC project. And we wanted to make sure that we have chemicals that cover a wide range of physicochemical properties, range of toxicities, and mode of toxic action. And another important point was that we wanted to have chemicals which were relatively easily quantifiable based on analytical protocols, which we had also established as part of the CEFIC project. Now, in terms of purchasing the chemicals, we first had the idea to buy it at the lead lab and then distribute. But again, due to the shipping difficulties, we very quickly realized this is impossible. And so we found a solution that we provided the information on a supplier, the lot number, the purity, so that each participant purchased the test chemicals themselves, but we all essentially had the same chemical then in hand. Another question that always comes up, do you need to code the chemicals? We decided we do not need this. And adding the difficulty with shipping the chemicals, it would actually not even have been possible in our case, because as I explained, everyone bought the chemicals themselves eventually. So what were the main organizational challenges for us? First of all, the study design. It's always easy to read these validation studies, but when you really get into the details, it's very difficult to find the information that you need. So we asked around a lot, we got different messages, and eventually we just made our own decision, hoping that what we decided on would be a valid study in the end. Having sustained resources was tremendously difficult to get. So first at the lead lab, obviously the validation study was just one of the many studies that we ran. We had to sort of prioritize this and set other research projects a little bit back in priority. And I must say, I'm really grateful for EAWAG that they said, we are in this, you go, you do this. But it really meant that we had to cut on other running projects. Same for resources for the participants. They do this validation as one of many of their tasks. And so we really had to respect the resource constraints, which meant, for example, that the study went on for much longer than initially anticipated. And as I've mentioned, we had change in personnel, specific safety rules. There's always something unexpected coming up, for which reason we had to adjust the validation program. So what were the main financial challenges? Well, I already explained that we had some kickoff fund, which was, however, insufficient. But overall, looking back, I think we were extremely lucky that the participants of our ring trial largely financed themselves, and that I was able to shuffle some institutional funds to keep the validation and guideline development running, obviously for years. So in conclusion, what factors were there that we were successful and what could be improved in the future? So I would say the main driver for success was our long-term commitment, and not only by us, but of course, obviously also by the participants. Funding situation needs to be badly improved. First of all, there should be easily accessible funding and realistic cost estimates based on the experience of successful validations. So if I would have to budget a validation study today, I think I would do much better than I did years ago when I had no idea what I was getting into. And we need to improve the validation process, in my view. So even though there are lots of documents, when we really looked for details, how to design the study, we were really lost. We couldn't find the necessary information. And I understand that this is a case-by-case evaluation, but guidance on this, someone to rely on who eventually is also responsible for standard and test guideline development would be really important. I imagine that there could be an experienced consultancy that can be put in charge of this kind of validation studies, while we need to keep in mind that there also resources are needed for the developer to train the other labs and obviously for the participants. So far to our experience, I'm very happy to give you further information. You can always contact me under the address provided here. Thank you very much.